Vanakkam. Welcome to this course on classics in neuroscience. In this video and in this series of videos, we have been looking at the biology of cell biology and the synapse. In this video, we will be looking at the chemical synapse and how the chemical synapse was first discovered, described, the contribution of Sanford Paley and the contribution of Eduardo de Robertis. So, what is this chemical synapse? It is a specialized junction through which neurons signal to each other and to non-neuronal cells, as in other excitable cells such as muscles or glands, for example. So, in this junction, the hypothesis is that some chemical is getting secreted or getting released, based on which the response happens in the postsynaptic cell. That can be a neuron, muscle or a gland or whatever, something. So, the junction between the neuron that is sending the information and the cell that is receiving the information, that junction is where this chemical transmission or this information sharing through chemicals is happening. This junction is called as the chemical synapse. This idea was heavily challenged by the reticularists, who said that there was no discontinuity, just that it appears like a discontinuity, but essentially neurons are connected throughout, a fully connected model. But this was not true. Any evidence that was earlier available before the 1950s were refuted by the reticularists, saying that there is still connectivity, there is still connection, there is still continuity. It required the advent of electron microscope to show that there is indeed a gap, that is the synaptic cleft, that is the region between the neuron that is sending the information and the cell that is receiving the information. There is a distinct space or a gap. This required electron microscopy because this kind of resolution was not possible in light microscopy, because there were serious optical limitations. So, the first electron microscopic images of synapses were reported in 1954 by Sanford Paley and George Palade in the United States, and simultaneously almost around the same time by Eduardo de Robertis and Bennett in Uruguay, approximately the same time. So, you are having these neurotransmitters that are packed in vesicles in the presynaptic cell, then this presynaptic cell fuses with the plasma membrane and then releases neurotransmitters. This process is called as exocytosis, we mentioned in the previous video, and then these are attached, these neurotransmitters attached to the receptors which respond. In this case, the particular case that we are discussing is the case of perhaps the case of a neuromuscular junction, but one has to check what it is. Here you are having some sort of ligand gated channel. So, there are specific channels that are there in this case. So, these receptors are sensitive to that chemical. Now, let us look at the work of Sanford Paley and his contributions to this field. Sanford Paley was born in 1918 in Cleveland. He received medical training at Western Reserve University. I think this is the university that is now called as Case Western Reserve University in Cleveland. He started working with Ernst Scharer and Beta Scharer on secretory granules in fish nerve cells, and he started to focus on electron microscopy much later, after he stopped working with or after he finished his training with Ernst and Beta Scharer. Much of his training was with Ernst and Beta Scharer, who were educated in Europe, Germany, and who brought with them this worldliness that they spoke many languages, they were very well aware of the literature, they knew authors of the paper that the students read, they knew authors of the textbooks. So, they were very well known academicians. So, Sanford Paley had the opportunity to work with this world class academicians. So, after finishing his work with Ernst and Betha Scharer, he took up a postdoc with Albert Cloud, and later he joined Yale in a teaching cum research position. He had a heavy teaching load, but it was the electron microscope that attracted him to Yale, because the access to electron microscope with the improvement in sectioning of tissues and visualization of tissues attracted him a lot. So, he joined Yale I think around 1949 and started focusing on using electron microscopy to study neuroscience. So, he started his collaboration with George Palade from about 1949-1950 onwards. I think George Palade was this refugee from Romania. After Soviet Union occupied Romania, George Palade went to the United States, and Sanford Paley started collaborating with George Palade to study the fine microstructure of the neuronal circuitry. So, they both used electron microscopy as their primary methodology, and their key discoveries was in the fine microstructure or the nanostructure of the neurons, identifying Nissl substance, endoplasmic reticulum, neurofilaments, Golgi apparatus. Parallelly, he also focused on identifying the synapse, and he studied motor nucleus or motor cells of the abducens nucleus and reported the first images of the synapses in 1954. Around the same time, de Robertis also reported this, but arguably Sanford Paley reported this set of work that he did on synapses in 1954, perhaps the first reported electron microscope images of synapses. Now, let us look at the work of Eduardo de Robertis and the contributions that he made to the field of neuroscience in general and to identifying the chemical synapse in particular. So, de Robertis was of Italian ancestry. He was born to Italian immigrants in Argentina in 1913. So, he trained in medicine, and he was very keen on doing biological research, and he got a fellowship to study in the US between 1939 and 1941, where he focused mostly on endocrine function. So, initially his interest was in endocrine function. So, after finishing his fellowship in 1941, he returned to Argentina and continued his research, and when Perón got to power in Argentina in 1946, he escaped from Argentina to go to the United States, where he studied with Frank Schmidt of MIT, the Massachusetts Institute of Technology, where they performed electron microscope studies of axons, and they demonstrated the presence of neurotubules. So, he was continuing that line of work. So, he was one of the early pioneers of the use of electron microscopy in studying neural structures. So, he used electron microscope to study axons. So, he was recruited by one of Ramon y Cajal's students to be part of the Biological Research Institute in Uruguay, So, he essentially was in a position to build the first electron microscopy facility in South America. So, there he performed some groundbreaking work or seminal, the very early work on the fine structure of retina. After that, he moved to the United States back once again. He collaborated with Bennett in Seattle in 1953, where he studied synaptic membranes and how exocytosis happens in adrenal medulla. This data he reported in 1954 in one of the first reports of the existence of synapse and synaptic membranes using electron microscopy. Later in 1957 he returned back to Argentina to continue his work in the cell biology department in Argentina. His contribution essentially amounts to the identification of synapses and synaptic membranes in adrenal medulla using electron microscopy, the perhaps one of the earliest reports of the existence of synapses. In this video, we looked at the chemical synapse, which is essentially the junction in which communication happens between one neuron and another cell, and the seminal contributions of Sanford Paley and Eduardo de Robertis, coming from two different backgrounds, and their contributions in terms of the identification of the synaptic membrane and the synapse using electron microscopy. With this, we come to the end of this lecture. Thank you very much for your attention.